Good morning to all of you. Here, today I am going to teach you one of the most important topic, that is cell organelle. And you know that in our body there are number of organs are present. Each and every organ they perform some specific function. For example, kidney, then brain, stomach, like that. Inside the cell, the number of cell organelles are present and they have the cell status like the organ, and they also perform some specific function. So today, among that, one of the cell organelle we will discuss in detail, and the name of that cell organelle is mitochondria. From ninth class, everybody learn about the mitochondria in detail. It is popularly known as the powerhouse of cell. So first we will check their details. So this mitochondria is one of the membrane bound. After that, semi-autonomous cell organelle. This is one of the membrane bound semi-autonomous cell replicating cell organelle present in the cytoplasm of almost all eukaryotic cells and totally absent in prokaryotic cells. In eukaryotic cell also, some of the cells are exceptional. For example, RBC. In RBC, mitochondria is absent. So this mitochondria is one of the most important cell organelle, and it is the largest cell organelle in animal cell and plays critical role to release energy. That's why it is popularly known as the powerhouse of cell. This mitochondria first time was seen by Gallagher in the year of 1880, and Zenga gave the name mitochondria in 1897 first time. So the word mitochondria is derived from two different words, that is mitos and chondrion. Mitos means thread like structure and chondrion means granule. So mitochondria is what? It is a granular thread-like structure present in the cytoplasm of almost all eukaryotic cells. This mitochondria, if you are check their morphology, so under the light microscope, when we are observe this mitochondria, so it requires specific stain first, and the name of that stain is Janus green. Through Janus green, it is seen under the light microscope, and the name of this stain is come from the person who developed that stain, which is green in color, and the name of the person is Janus. That's why it is well known as the Janus green stain. Now we will check the morphology of mitochondria. This mitochondria, it was checked under the light microscope. It generally shows cylindrical in outline. If you are checking their shape, generally cylindrical in outline. However, it also shows some other shapes, right? Means branch also, then sausage shape. These are the different shapes are also present in mitochondria. But the shape of mitochondria is controlled by the physiological condition of the cell, activity of the cell. Right? So This is the first character morphological. Second, their size. If you are check the size of mitochondria is about 1 to 4.1 micron in length and 0.2 to 2 micron in diameter is the size of mitochondria. Now their number after the same size in morphology we will take the number also. Number shows great variation. In some cells only one mitochondria is present. For example, in chlorella only one mitochondria is present. It is unicellular organism. Then in case of our liver cell, if you have checked, that is 500 to 1000 mitochondria are present. Then in case of oocytes, there are 30,000 for within one cell. Then in giant mitochondria, giant amoeba, there are 50,000 in one cell. And in case of flight muscle cells of insect, there are 5 lakh in number. In flight muscle cells. So this indicates that the number from 1 to up to 5 lakh, this indicates that the number of mitochondria is depends on activity of cell, type of cell or function of cell. So here we just check in plural they are the unicellular, they show less locomotion. In case of liver in our body, then in oocytes, in giant amoeba, means what? The activity of cell is increased, the number of mitochondria is increased. If you are checked in plant cell and animal cell comparatively, so in plant cell, the number of mitochondria is less than the animal cell because the metabolic activity of animal cell is higher than the plant cell. This is the first reason. Second, animal shows locomotion. That's why they require more energy. That's why the number of mitochondria is more in animal cell than the plant cell. And plant cell is show sedentary nature, means they not show locomotion. They show only movement. So whole lifespan they complete at one place. So in that locomotion is absent. So energy level is also less. That's why the number of mitochondria is less in plant cell than the animal cell. Even the condition of that organ or the cell is also determine the number of mitochondria. For example, in case of dormant cells and germinating cells, if you are compared, in both the sites the number of mitochondria is different. In dormant cells, the number of mitochondria is less, while when the seeds start to germinate, the metabolic activity is increased and the number of mitochondria also increase. Right? So in this way, shape, size and number of mitochondria shows great variation. On the basis of that, the type of cell, activity of cell and physiological condition of cell, the number of mitochondria is more or less. Right? Now, we will take the details of mitochondria. So, it is necessary to study their ultrastructure because without ultrastructure, we are not understand means what is the actual function of mitochondria. Right? So, what do you mean by ultrastructure? The ultrastructure of any cell organelle means what? When we are studying this organ under the electron microscope, with high magnification, that is called as the ultrastructure. So, through the ultrastructure, we come to know the several functions of mitochondria and what is present and what are the events take place inside the mitochondria, why it is important, And especially when we are studying the respiration, cellular respiration, how substrates are oxidized inside the cell or inside the mitochondria. For that, ultrastructure is very important. According to the ultrastructure of mitochondria, this mitochondria shows two membrane and two chambers. I repeat, two membrane and two chambers according to the ultrastructure. Ultrastructure of mitochondria. According to that, so it has two membrane. In introductory part also we have discussed. It is membrane bound cell organelle like this. This is the outermost membrane which is permeable to number of metabolites and it allows entry of various type of metabolites. It is highly permeable. This is outer membranes. But here the enzymes, if I check, the number of enzymes are less. Protein is also less, but the lipids are more. Cholesterol, this type of lipids are present more in outermost membrane. While in comparison of outer, the inner membrane is somewhat different. If the outer membrane is continuous, but inner membrane is folded, it shows number of folds like this. It is enfolded. Like this, number of folds are present that increase the surface area for physiological activity like that. Right? And each fold is called as a cristae, like this. Suppose this one. This each fold is called as a cristae. So this cristae or their number indicate the intensity of respiration. If the number of cristae is more, this indicates the more respiration is done inside the mitochondria, and the number of cristae is also variable. Means it depends on requirement of energy. Depends on requirement of energy and depends on type of cell also. In which cell the mitochondria is present, on the basis of that, here the cristae or their number is increased or decreased. So this outer membrane and inner membrane, they have number of different cells. That is inner membrane. As you see that outer membrane is continuous while the inner membrane is folded. This is the first difference. Second difference is what? Outer membrane is highly permeable to number of metabolites while the inner membrane is somewhat selectively permeable. Outer membrane has less number of enzymes while the inner membrane has maximum enzymes that are present inside the inner membrane and that contribute into the oxidation of substrate. This outer membrane has less number of proteins while the inner membrane has number of carrier proteins. This is the difference. Outer membrane has more lipids while the inner membrane has less lipids. So these are the number of differences between outer and inner membrane of mitochondria. Right now, the side of inner membrane toward the inner chamber. Because of these two membranes, here two chambers are formed. This is outer chamber, like this. That is outer chamber. The space between two membranes is called as the outer chamber. It is extend up to the cristae, this much, right? And inside the inner membrane, the proteinous jelly-like substance is present. That is called as the matrix. This matrix is present in the inner chamber. This inner chamber is filled with the matrix. Right? So, according to the ultrastructure, mitochondria has two membrane and two chamber. So, outer membrane, inner membrane, these are two membranes, and chambers are the outermost chamber is present in between the outer and inner membrane. That is also called as perichondrial space. It is also called as the perichondrial space. While inside the inner membrane, the core of mitochondria is present. That is called as the inner chamber, which is filled with the proteinous jelly-like substance or material. That is called as the matrix. Now, first we will check the inner membrane. The side of inner membrane toward the inner chamber is called as the end site. Means inside the inner membrane toward the inner chamber, this side is called as the end site, and the side of inner membrane toward the outer chamber is called as the C site. So this is the C site and this one is the end site. Now inside the inner membrane here, tennis ball like number of structure are present that we call as the F1 particle or oxygome. These oxygomes are present in each cristae or inner side of inner membrane, like that. On each cristae, such types of pinhead-like structure are present, like this. So this cristae increase the physiological active surface area. Now we will check separately how the cristae is formed, like that. That we call as a cristae, and on each cristae number of this oxygome is present. Each oxygome is globular head, middle stalk, and the base, that is head, middle stalk, and this one is the base, that is oxygome. It is also called as the F1 particles. So inside the inner membrane, here such types of pinhead-like structure are present. This structure, folded structure, is called as a cristae, and inside the cristae, inner membrane of cristae, the number of F1 particles or oxygomes are present. Each oxygome having globular head, middle stalk, and the base. This base having ATPase enzyme. That's why this F1 particle or oxygome is acts as a center for release of or synthesis of ATP. Synthesis of ATP during the oxidative phosphorylation. Means when the food or the respiratory substrate is oxidized, that time ATP are synthesized due to the presence of ATPase in the base of oxygome or F1 particle. Now inside the inner membrane, here we are checked that is proteinaceous gel-like substance is present, that is called as the matrix. So this matrix are present in the inner chamber of mitochondria, that is the core of mitochondria. Inside the matrix, the number of substance is present. The most important is DNA. In eukaryotic cell, only two cell organelles apart from nucleus, they have their own DNA. In that, chloroplast is one in case of plant cell, and second cell organelle, that is the mitochondria. Only these two cell organelles having their own DNA. That's why they both are well known as a semi-autonomous cell organelle. Due to presence of this DNA, it shows autonomy means they replicate itself due to presence of DNA. They synthesize their own structural protein. They synthesize their own RNA, mRNA, tRNA, rRNA. Such types of RNA are synthesized inside the mitochondria due to presence of DNA. So they have their own DNA. Second one is ribosome. Mitochondrial ribosome is shows similarity like the prokaryotic ribosome. It is just like a 70S type of ribosome. 50 to 70S type of ribosomes are present in the inner chamber of mitochondria or the matrix of mitochondria. The most important thing that is respiratory enzymes, which is necessary for Krebs cycle. So when the food substrate is entered into the Krebs cycle, means that is the pyruvate, after the acetylation when it enter into the Krebs cycle, then it is converted into number of intermediate products, and in each step they require specific enzyme. All that enzymes are present in the matrix of mitochondria. After that, apart from this, they have their own proteins also and other raw material required for cellular respiration. Right? So in this way, this inner chamber, after that outer chamber, two chambers are there according to the ultrastructure of mitochondria, and two membranes, outer membrane and inner membrane. After the inner membrane, the outer membrane is extend up to the cristae, up to the inner membrane. Here, this outer membrane has jelly-like substance, but it is less number of enzymes, and it is called as the pericordial space. When we are studying in 11th class, when you will learn about the respiration, cellular respiration, that time you come to know what is the importance of pericordial space, after that the matrix, because the glycolysis in cellular respiration there are three main steps. When the food substrate is completely oxidized, the first step is glycolysis. It takes place in cytoplasm of the cell. Suppose this black body you consider as a cell, right now, and that is the cytoplasm. So glycolysis takes place in cytoplasm. At the end of the glycolysis, the product is got per glucose molecule, two pyruvates are formed. Then that pyruvate enter into the pericordial space in case of eukaryotic cell. So this pericordial space is the site of acetylation. Here, that pyruvate convert into acetyl-CoA. Then acetyl-CoA is enter into the matrix where the Krebs cycle is take place, and after the Krebs cycle, then electron transport system is present in the inner membrane of mitochondria, and at the base of F1 particle ATPs are present. So oxidative phosphorylation is take place near the F1 particle, at the base of F1 particle. So this inner membrane has the number of essential enzymes required for ETS. So glycolysis takes place in the cytoplasm of cell. After that, acetylation takes place in the pericordial space of mitochondria. Krebs cycle takes place in the matrix of mitochondria, and ETS is take place at the base of F1 particle, right now, or inner membrane where all the enzymes are present. Because when we are comparing between inner and outer membrane, so inner membrane is rich with the number of enzymes as compared to the outer membrane. Right now. So each and every substance present in the matrix, they increase the efficiency of mitochondria, especially the DNA, that make the mitochondria autonomous. But it is not fully autonomous. Partially, there some function is controlled by the nucleus. Right now. So that's why mitochondria is called as the semi-autonomous cell organelle present in almost all eukaryotic cells. This is all about the ultrastructure. Now we will check the function of mitochondria. As we have discussed before, so mitochondria is considered the biochemical factory, because while oxidation of food substrate, respiratory substrate, the number of intermediate products are formed that is necessary for the synthesis of chlorophyll, steroids, amino acids, right? Even the first amino acid, glutamic acid or aspartic acid, are synthesized during the Krebs cycle through the alpha ketoglutarate acid, or from that alpha ketoglutarate acid and oxaloacetic acid, respectively. So, the number of intermediate products are formed during the Krebs cycle inside the mitochondria. Second thing, this site where the food substrate is completely oxidized, and at the end, the number of ATPs are released, and that ATP is further utilized for activity or metabolic activity of the cell. That's why this mitochondria is considered as the powerhouse of cell. Right? So this mitochondria is provide energy also. It provides number of intermediate products also. That is further used for the synthesis of number of biochemicals, biomolecules like chlorophyll, steroids, amino acids, like that. Right? So these are the function of mitochondria. So in this lecture we are discuss what is meant by mitochondria. So mitochondria is one of the membrane-bound cell organelle. It is one of the self-replicating semi-autonomous cell organelle present almost in all eukaryotic cell cytoplasm and totally absent in prokaryotes. Right now. Then we are also checking their morphology, shape, after that size, their location, their number is depends on mostly the type of cell, activity of cell, and physiological condition of the cell. So these are the different characteristic features, morphological features we are discussed. And according to the Alta structure, mitochondria is two membrane and two chamber structure. Both the membranes they show number of differences. Outer and inner membrane. If outer is continuous, the inner membrane is folded. Outer is mostly permeable, freely permeable, but inner membrane is allow entry and exit of few metabolites. Right now, outer membrane has less enzyme, but inner membrane rich with the enzymes. Outer membrane has less protein, inner membrane has more carrier protein. Outer membrane has maximum lipids in the form of cholesterol, but inner membrane has less number of lipids. So these are the differences. But then also there is some adhesive portion, means where both the membranes are come close into their contact, and through that point they exchange the material between the outer membrane to inner membrane and inner membrane to outer membrane. There is some point also present. So inside the inner membrane, what is present? How it formed that cristae? Then each cristae having number of pin-head-like structure that we call as the F1 particle or oxygen. Each oxygen having globular head, middle stalk, and base. And the function of that F1 particle is what? At their base, the ATP synthesis are present that contribute in the synthesis of ATP during the oxidative phosphorylation. So, inside the inner membrane, there is one chamber, and outside the inner membrane or the space between the inner and outer membrane, the outer chamber is present. Outer chamber is extended up to this gap, and it has less number of enzymes, while the inner membrane having maximum enzymes that contribute to the Krebs cycle, and these are the substance present inside the inner membrane that we call as the matrix. And due to presence of DNA, how mitochondria is perform some function autonomously, and that's why it is called as the semi-autonomous. And the function of mitochondria we already discussed, means it is the powerhouse of cell, because here the number of ATP are synthesized during the oxidative phosphorylation, and that ATP they utilize for the